This is an assertion reasoning question. The life cycle of Rhizopus is haplontic. The zygospore is the only diploid structure that shows diploid state in the life cycle. Because zygospore is the only diploid structure, Rhizopus life cycle is haplontic. So, both A and R are correct. R is the correct explanation of A, first option. A plus gamete angium of Rhizopus with 150 nuclei fuses with minus gamete angium having 100 nuclei. What is the diploid number of nuclei? 150 in plus, 100 in minus. So, diploid nuclei will be 100. Diploid nuclei will be 100. Degenerated haploid nuclei will be 50. Degenerated haploid nuclei will be 50. Then, what is the number of diploid nuclei? 100. Then, haploid nuclei present in germinating zygospore will be 400. So, answer is 100 into 4 is 400. Answer is 100, 50 and 400. Second option, not third option, it is second option, 100, 50 and 400. Meiosis occurs in after germination of zygospore is wrong, after formation of zygospore is wrong, before germination of zygospore is correct, after dormancy is also wrong. So, in Rhizopus or anywhere meiosis occurs before the germination of zygospore, haploid nuclei are formed. In Spirogyra, 3 nuclei disintegrates. In Rhizopus, all the nuclei remain functional and produce germ spores. Third option. Arrange the following in a sequence. Arrange the following in a sequence. First, always trisporic acid, then zygophore. So, after the activity of trisporic acid, zygospore is formed. Then progametangium, every zygophore produces progametangium. Then progametangium divides into 2 by a septa. Lower portion is called suspensor, upper portion is called gametangium, then gametangium. Then finally, after gametangium, reduction, division and promycelium. Reduction, division in zygospore and finally progam. So, trisporic acid, zygophore, progametangium, gametangium, reduction, division, promycelium. BEFACD is the correct answer for this question. Second option. The hyphae which grow in opposite directions. In Rhizopus, the hyphae which grow in opposite directions, these are stolons and these are sporangiophores. These are stolons and these are rhizoids and these are sporangiophores. This is sporangiophore and this is sporangia. So, the rhizoids, stolons and sporangiophores all formed during reproductive stage. What are the structures which are growing opposite to each other? Means rhizoids grow into the medium, sporangiophore grows opposite to the medium. So, definitely rhizoids and sporangiophores are the structures which grow in opposite directions. So, the stolons and sporangiophore does not grow in opposite direction. Stolons and zygophore does not grow in opposite direction. Stolons and rhizoids does not grow in opposite direction. So, what are the structures that are growing in opposite directions? Means rhizoids and sporangiophores. Second option. A plus gametangium has 80 nuclei while a minus gametangium has 80 nuclei, plus gametangium has 100 nuclei. How many nuclei are present in resting and germinating zygospore result after the fusion of such gametangium? Whenever minus has 80 and plus has 100, only 80 will fuse. Nuclei fuse in pairs, unpaired nuclei disintegrate is the concept in Rhizopus. So, 80 diploid nuclei will be there. So, resting zygospore contain 80 nuclei, germinating zygospore contain 320 nuclei, because 80 into 4, 320. Answer is second, 80. Arrange the following events in a sequence. Absorption of digested food material, secretion of enzymes, fixation and formation of sporangiophores. First, fixation with the help of rhizoids and sporangiophores. Then secretion of enzymes. Rhizoids start secreting exoenzymes which depends or which acts on the medium, digests the medium. So, secretion of enzymes. Then absorption of digested food material, that is A. Then formation of sporangiophores. So, first what happens? First stolons are formed. Then rhizoids are formed. Then rhizoids secrete enzymes. Then sporangiophores are formed. food material move to the tip and ultimately sporangium is formed. So, first secretion of enzymes from rhizoids. Then first is fixation. It has to be fixed with the help of rhizoid. Then secretion of enzymes. Then absorption of digested food material. Finally formation of sporangia. These are all the stages which occurs in a sequence during asexual reproduction in Rhizopus. So, C, B, A, D will be the answer. Rhizopus sexualis is homothalic species. It undergoes sexual reproduction. Rhizopus sexualis is homothalic species is correct. It undergoes sexual reproduction is correct, but it is not given whether it is self-compatible or self-incompatible. It is actually self-compatible where single mycelium is enough to complete the reproduction. So, it is not a clear explanation for reason. Reason is not the correct explanation of assertion, but it is true. It undergoes sexual reproduction. So, as both are correct, one is not related to other. The answer will be both A and R are true. R is not the correct explanation of A. Animal characters found in Rhizopus. Animal characters found in these are all saprophytic fungi which grow on dead and decaying organic matter, particularly on a medium rich in glycogen they grow. Animal character in Rhizopus is extracellular digestion. Enzymes are secreted and medium is digested. Absorption of digested food material from outside, it is wrong. Digested food material is not taken by animals. Absence of chlorophyll, it is also correct. Absence of chlorophyll is also correct. Conjugation occurs in Spirogyra and other ones also. Gametangial copulation is correct. So, answer will be gametangial copulation will not be, only conjugation. So, extracellular digestion, absence of chlorophyll, conjugation 1, 3, 4 are the animal characters normally found in a Rhizopus. The following must occur during the formation of sporangiospore: mitosis, meiosis, cleavage, dehiscence. Answer is cleavage. Why? Because once sporangium is formed, columella is differentiated, then cleavage must occur in sporangium, so that first irregular chambers are formed, but every irregular chamber next converts into circular specialized spores called eplanospores or sporangiospores. Without cleavage, no question of irregular chambers. Without irregular chambers, no chance of formation of circular spores. So, the following events must occur during the formation of sporangiospore means cleavage. Cleavage must occur. Even mitosis does not occur, even meiosis does not occur. Dehiscence definitely occurs at maturity, but what should be definitely occur during the formation of sporangiospores in the sporangium of asexual reproduction is cleavage. Cleavage must occur. So, 3 only. The thick-walled resting spores echinids formed in Spirogyra farlovi are. Just now we discussed echinid is formed from the total cell wall and protoplasm. So, it is uninucleate and diploid is wrong. Cenozytic haploid is wrong. Cenozytic diploid is wrong. It is simply uninucleate and haploid. So, the thick walled resting spore echinid formed in Spirogyra farlovi are uninucleate and haploid. First option. Non-resting spores of Spirogyra are. Non-resting spores of Spirogyra. Parthenospores are non-resting or resting spores. Zygospore is a resting spore. Echinid is resting spore. Why? Because these are all surrounded by thick layered. What are parthenospores? Non-functional eplanogametes. Sometimes get surrounded by thick wall and converts into parthenospore. So, they are thick walled means taking rest. Zygospore also has 3 layered thick wall around it, definitely resting spore. Echinids are also thick walled spores helping in rest, performs rest period. Eplanospores are not surrounded by that thick wall, they get separated from the cell and develops into new Spirogyra septate hyphae. So, non-resting spores of Spirogyra are eplanospores, third option. The ratio of conjugating cells, zygospores and new filaments formed in Spirogyra. The ratio of conjugating cells always 2. The ratio of conjugating cells always 2. Any conjugating cell, 1 zygospore is formed. Each zygospore produces 1 new filament. So, the ratio of conjugating cells, zygospore and new filaments is 2:1:1. 2:1:1. Conjugating cell number will be always 2. Either it is a scalariform conjugation or lateral conjugation. In scalariform conjugation, 2 cells of 2 filaments. In lateral conjugation, 2 cells of the same filament involved. Zygospore formed if 2 cells involve, only 1 zygospore is formed. And new filaments, every zygospore, even though it shows meiosis and produce 4 haploid nuclei, 3 disintegrate, only 1 is functional. So, every zygospore produce only 1 new filament. So, 2:1:1. The male gametes of Spirogyra jogensis differ morphologically from female gametes in having. In Spirogyra jogensis, direct lateral conjugation was reported in MOP Iyengar. In Spirogyra mirabilis, it was reported by R.S. Rathan. What its question means? In both the cells, the cell above the holdfast cell is female, above the female cell is male cell. The protoplasm unites to form aplanogametes, both aplanogametes. But in male, female cell increases its size, but male cell does not increases its size. In male cell, if you clearly observe, the aplanogamete produces a small outgrowth or projection called conical process, which pierces through the septa and fuses with other cell. So, the male gametes of Spirogyra jogensis differ morphologically from female gametes in having a conical process. This conical process is completely absent in female gametes. So, answer is 4. If one of the conjugating gametangia of Rhizopus has 120 nuclei and other has 100 nuclei, and a plus is having 120 nuclei, minus is having 100 nuclei. The number of nuclei that would be present in germinating zygospore. Germinating zygospore means meiosis is over. So, one has 100 nuclei, other has 120 nuclei. 100 nuclei fuse, because nuclei always fuse in pairs. Unpaired nuclei disintegrates. How many nuclei disintegrates? 20 disintegrate. So, 100 diploid nuclei will be there, and these 100 diploid nuclei undergo reduction division and produce 400 haploid nuclei, which later form 400 meiospores, where 200 are plus spores and 200 are minus spores. So, answer for this is 400. 400 is the answer. Conjugation tubes are formed during the conjugation in all Spirogyra species. Amoeboid gametes of most of the Spirogyra species move through conjugation tubes. Conjugation tubes are formed during conjugation in all Spirogyra species is wrong, because in Spirogyra jogensis, where direct lateral conjugation takes place, no question of formation of conjugation tubes. But amoeboid movements of most of the Spirogyra species move through the conjugation tube is correct. Even in isogamous, amoeboid movements takes place of both the gametes. So, here A is false. Conjugation tubes cannot be formed during the conjugation in all species of Spirogyra is wrong. Amoeboid gametes of most of the Spirogyra move through the conjugation tube is almost correct. Most of the Spirogyra is correct. So, answer A is false, R is true. A is false, R is true. The conjugating filaments of Spirogyra affinis are considered as monoecious filament, definitely, because 2 cells of the same filament involve in the indirect lateral conjugation. In 2 adjacent conjugating cells of Spirogyra affinis filament, one acts as male and another acts as female. One acts as male and other acts as female. So, the conjugating filaments of Spirogyra affinis are considered as monoecious filaments is correct. In 2 adjacent conjugating cells of a Spirogyra affinis filament, one acts as a male and other acts as a female. Once they act as male and female, then only conjugation takes place. So, both are related to each other. Definitely the answer will be both A and R are correct. R is the correct explanation of A. When plus gametangium of Rhizopus with 50 nuclei minus gametangium with 52 nuclei of Rhizopus participate in conjugation, then dormant zygospore contain 50 nuclei of plus, 50 nuclei of minus. So many questions are given in this, because this is a very important concept, definitely asked in EAMCET. So, when a 50 nuclei of plus and 50 nuclei of minus fuse and 2 nuclei disintegrates, dormant zygospore has 1 or 2 haploid nuclei is wrong. 50 pairs of haploid nuclei is wrong. 200 haploid nuclei forms after the dormancy. So, here question asked is dormant zygospore. So, answer is 50 diploid nuclei. Dormant zygospore contain 50 diploid nuclei. When plus gametangium with 50 nuclei minus gametangium with 52 nuclei participates in copulation and produces the zygospore. And dormant zygospore definitely has 50 diploid nuclei. So, fourth is the option. The mating types of heterothallic species of rhizophore are indistinguishable. Indistinguishable. Rhizopus stolonifer is heterothallic species. This heterothallism in Rhizopus stolonifer was proposed by A.F. Blacksley. Here one is a plus mycelium, the other is a minus mycelium. Plus mycelium cannot involve in reproduction, minus mycelium cannot involve in reproduction. Only plus and minus, when they come close to each other, then only they involve in reproduction. So, this process is nothing but physiological anisogamy. Why? Both are morphologically similar but physiologically dissimilar. One is plus, the other is minus. Rhizopus stolonifer is a heterothallic species. Heterothallism is in Rhizopus stolonifer was explained by A.F. Blacksley. He proposed that heterothallism is the starting stage of development of dioecious condition in higher plants. That means in future plants are going to develop with male plant and female plant separately. So, the mating types of heterothallic species of Rhizopus are indistinguishable, means we cannot recognize them. Physiologically we can recognize them. Physiologically and morphology is wrong. Ever in the life cycle is wrong. Morphologically we cannot indistinguish, that is we cannot distinguish them morphologically. By seeing them we cannot say that which one is plus and which one is minus. Only by reproduction we came to know that they are, one is plus, the other is minus, because 2 plus cannot involve, 2 minus cannot involve. Plus and minus when kept together they involve in reproduction and produce zygospores. So, second option. Rhizopus can be called as pin mold after the formation of. When Rhizopus is growing like stolons above the medium like white cottony mass, then it is called bread mold. When Rhizopus produces sporangiophores opposite to rhizoids and then small bulged sporangium at the tip of sporangiophore, it resembles like a pin, hence it is called pin mold. When it produces spores, when these spores fall on the medium, the total medium converts into black color, hence it is also called black mold. So, various names of Rhizopus. Because the stolons grow extensively over the medium, it is bread mold. Because of the formation of sporangiophore and sporangia resembling like a pin, it is pin mold. Because black colored spores are formed and the medium converts into black color, it is black mold. So, Rhizopus can be called pin mold after the formation of asexual sporangia, not by chlamydospores, not by zygospores, not by germ sporangia. Germ sporangia form rarely, but asexual sporangia form in clusters, large number, which resembles a pin, that is called as pin mold. Identify the character that can be used to distinguish Thallophyta from other divisions of plant kingdom. Presence of cellulosic cell walls is common in all algae, fungi, bryophytes, pteridophytes, gymnosperms, everything. Reserve food metal is usually starch. In algae it is starch, in bryophytes may be starch, in pteridophytes may be starch, in gymnosperm is starch, in angiosperms is starch, because all are green in color, perform photosynthesis. But in fungi it is glycogen and oil globules, because it is heterotrophic thallophyte. Absence of vascular tissues. Absence of vascular tissues can be observed even in bryophytes. Bryophytes also are atracheophytes along with thallophytes. The only difference between the thallophyte and other divisions of this is absence of embryo, because Thallophyta is the only group where the zygote undergoes mitosis instead of meiosis. It undergoes meiosis instead of mitosis. Whenever it undergoes meiosis instead of mitosis, embryo is not formed. So, identify the character that can be used to distinguish Thallophyta from other divisions of plant kingdom. Other divisions means Bryophyta, Pteridophyta, Spermatophyta. Spermatophyta has angiosperms and gymnosperms. So, presence of cellulosic cell walls we cannot say. Reserve food material is usually starch, we cannot say. Absence of vascular tissues also okay, but the striking feature of all thallophytes is absence of embryo, because zygote undergoes meiosis in the place of mitosis. So, absence of embryo is the striking feature that distinguishes Thallophyta from other divisions of plant kingdom. In the life history of bryophytes, the dominant phase is gametophyte, but sporophyte is independent. Wrong. The dominant phase is gametophyte is correct, but sporophyte is independent is wrong. The dominant phase of gametophyte and the sporophyte is either complete or partial parasite on the gametophyte is correct. So, dominant phase is definitely gametophyte. Sporophyte is complete or partial parasite on gametophyte. The dominant phase is sporophyte itself is wrong. The dominant phase is sporophyte is again wrong. And this gametophyte is very much reduced and totally parasitic, and sporophyte is angiosperms. The dominant stage is sporophyte, but the gametophyte is independent, definitely pteridophytes. The dominant phase is gametophyte, but sporophyte is independent is not there. So, dominant phase is gametophyte and the sporophyte is complete or partial parasite on the gametophyte. So, in the life history of bryophyte, second option is clearly found. What is that? The dominant phase is gametophyte and the sporophyte is either partially or completely depending on gametophyte. So, second option is the correct answer for life history of bryophytes. Arrange the following plant groups in ascending order with reference to their gradual reduction of gametophytic stage. That means gametophyte slowly starts reducing and sporophyte becoming prominent. That is the question. Gametophyte is reducing slowly, sporophyte is becoming prominent. So, gametophyte is very much found in bryophytes. Next, gametophyte is reduced and sporophyte a little bit prominent in pteridophytes. Then sporophyte almost reduced, almost sporophyte is very much developed, and gametophyte also appeared in the case of gymnosperm. Gametophytes are fully reduced and completely parasitic on sporophyte in angiosperm. So, answer will be second one, Bryophyta, Pteridophyta, gymnosperms, angiosperms. So, what is this question? Arrange the following plant groups in ascending order. Ascending order means from lower to higher. That means gametophyte is present and gametophyte is reduced. So, Bryophyta, Pteridophyta, gymnosperms, angiosperms are the plant groups where gradual reduction of gametophytic stage is clearly observed. 6 Spirogyra filaments, 6 Spirogyra jogensis filaments with 101 cells each participate in monoecious conjugation. A number of young filaments produced from the zygospores. 6 Spirogyra filaments with 101 cells each. Forget about the cells present in the filament, because Spirogyra jogensis shows direct lateral conjugation where only 2 cells involved from the base. Above the holdfast female cell, above the female cell, male cell. So, 6 Spirogyra filaments involving means 6 zygospores are formed. If 6 zygospores are formed, the number of young filaments produced will be 6. So, 6 Spirogyra filaments with 101 cells each participate in monoecious conjugation. Number of young filaments produced from the zygospores are only 6. 300, 600, 60, just to confuse. 6 Spirogyra jogensis means we have to remember Spirogyra jogensis is direct lateral conjugation where 2 cells involve. The cells above the holdfast female, above the female is male. So, 6 zygospores are formed. Each zygospore produces only 1 young filament. So, how many young filaments are produced means only 6. So, answer is definitely 3. Next. Consider the following statements with regard to Spirogyra and identify which one are correct. Each cell has 1 to 16 chloroplast in periphery. Correct. During the vegetative reproduction, each fragment grows into full filament by apical growth. Apical growth is wrong. Apical growth is always wrong. The growth in Spirogyra is called diffused. Every cell is having capacity of undergoing transverse division and producing new filament. Then, Echinite is a resting pore formed due to the contraction of protoplast is wrong. Whenever echinite formation occurs, total cell involves, and echinite is formed by the complete involvement of cell wall and protoplast. So 2 and 3 are definitely wrong. In monoecious conjugation, conjugation tube may be formed or may not be formed is correct. Why? Monoecious conjugation is of two types: direct lateral conjugation, indirect lateral conjugation. Direct lateral conjugation, conical process is formed in the male gamete, and the male gamete pierces through the septa at the center, enters the female gamete and GM and fuses with the aplanogamete there. May or may not be. May be formed is in Spirogyra affinis, where indirect lateral conjugation takes place. Lateral conjugation canals are formed on the lateral sides. So, lateral conjugation canals in Spirogyra affinis. Spirogyra affinis, indirect lateral conjugation. All the cells in the filament involves. On the sides, first papillate outgrowths develop, and this cell contents condenses to form aplanogametes. The cell with aplanogamete is called gametangium. These will grow and ultimately fuses to form lateral conjugation canal. Lateral conjugation canal. So, in Spirogyra affinis, lateral conjugation canal is formed. In Spirogyra jogensis, conjugation canal is not formed. So, fourth option is in monoecious conjugation, conjugation tube may be formed or may not be formed. May be formed is indirect, may not be formed is direct. Echinite is a resting spore formed due to the contraction of protoplasm is wrong. So, answer for these 1 and 4. Each cell has 1 to 6 chloroplast in periphery is correct. Apical growth is always wrong. Apical growth occurs in Rhizopus, not in Spirogyra. In Spirogyra, the growth is popularly called diffused growth because every cell undergo transverse division. So, 1 and 4 are correct. Each cell has 1 to 16 chloroplast in the periphery. In monoecious conjugation, conjugation tube may form or may not be formed. So, 1 and 4 are correct. Third is the option. Arrange the different events in order during the formation of zygospore of Spirogyra. First is always germ tube. First germ tube, then meiosis, then diploid nuclei, then haploid nuclei. So, answer is 4. 3, 1, 2, 4. 3, 1, 2, 4. Germ tube, first meiosis, then first diploid nucleus 2, then meiosis 1, then haploid nuclei, then germ tube. Answer is not 4, it is 2. 2, 1, 4, 3. The spore produced in the germ sporangium of Rhizopus stolonifer on germination gives 2. The spore produced in the germ sporangium of Rhizopus stolonifer germination. Germ sporangium, the spore formed in germ sporangium either plus or minus. Mycelium, which is multinucleate but all the nuclei of the same strain. Mycelium which has uninucleate cells, wrong. Mycelium which are multinucleate but nuclei of two different strains. Mycelium in which alternate cells are having the nuclei of two different strains. So, the spore produced in germ sporangium of Rhizopus stolonifer on germination gives rise to mycelium which is multinucleate but all the nuclei are of same strain, because whether it may be a plus strain plus spore or may be a minus spore. If it is a plus spore, all plus mycelia. If it is a minus 4, all minus mycelia. So, answer is mycelium which is multinucleate but all the nuclei are of the same strain. Arrange the following events in descending order: promycelium, zygospore, zygophore, gametangium. So, first zygophore, then gametangium, then zygospore, then promycelium. 3, 4, 2, 1. 3, 4, 2, 1 is the answer for this. What is the minimum number of nuclei that might have been present in the plus gametangium of a Rhizopus species when the resulting germinating zygospore has 200 nuclei? So, resulting zygospore has 200 haploid nuclei. We have to do it in a reverse order. 200 haploid nuclei means 50 diploid nuclei. 200 haploid nuclei means 50 diploid nuclei. 50 diploid nuclei means 50 nuclei in each gametangium. 50 haploid nuclei in each gametangium. When 50 haploid nuclei in each gametangium fuses, then by gametangial contact, if 50 haploid nuclei fuses by gametangial contact, 50 diploid nuclei are formed. 50 diploid nuclei undergo reduction division and produce 200 haploid nuclei. So, what is the question here? What is the minimum number of nuclei that might have been present in the plus gametangium of a Rhizopus species when the resulting germinating zygospore has 200 nuclei? If it is 25 diploid nuclei are formed and 100 haploid nuclei are formed. If it is 100, 400 are formed. If it is 800, 3200 might have been formed. So, if it is only 50, 50 plus 50, 50 haploid of plus and minus becomes 50 diploid, and 50 diploid into 4 becomes 200 haploid nuclei. So, answer is 2. Match the following: cytase action, larger female gamete, a zygospore, zygospore in conjugation canal, parthenogenesis, Spirogyra graciana, isogamy, anisogamy, division of labor. Now, cytase action A, Spirogyra graciana, because it shows both scalariform and lateral conjugation. Larger female gamete, anisogamy, that is not anisogamy, it is 4, anisogamy. Azygospore is parthenogenesis. What is parthenogenesis? The unfused eplenogamete sometimes get surrounded by thick walled spores around them, thick walled layers around them, resulting in the formation of a zygospore. Then zygospore in conjugation canal, isogamy. Whenever isogamy takes place, the eplenogamete of this cell, plus or male or whatever it may be, the eplenogamete of this cell both will come and ultimately fuse to form zygospore in conjugation canal. So, cytase, for this answer is 2. Cytase action 2, Spirogyra graciana. Larger female gamete 4, anisogamy. Azygospore parthenogenesis 1. Zygospore in conjugation canal, isogamy. So, A2, B4, C1, D3 will be the answer for this. Now, match the following: trimorphic conjugation, dimorphic cells, dimorphic gametes, sexual spore, zygospora, Spirogyra jogensis, Spirogyra adnata, Spirogyra happinis and Spirogyra firelove, whatever it may be. Answer is 3. A4, trimorphic combination is Spirogyra graciana. Spirogyra graciana is missed there. Trimorphic because it shows both scalariform and lateral conjugation. Dimorphic cells is Spirogyra jogensis. Why? 2 cells, male cell and female cell. C1, dimorphic gametes, zygospore, and C2, Spirogyra jogensis, dimorphic gametes, because one male gamete shows a conical process, female gamete does not show any conical process. So, Spirogyra jogensis is dimorphic gametes, and D1, sexual spore is zygospore. So, trimorphic conjugation, Spirogyra graciana 4. Here graciana is missed, Spirogyra graciana. B3, dimorphic cells is Spirogyra adnata, because 2 cells in the same filament help in that. Dimorphic gametes is Spirogyra jogensis, conical process in male gamete and non-conical process in female gamete, and D1, sexual spore is zygospore. So, A4, B3, C2, D1. Now, this is a 3-column tabular form. Type of spores, nature of wall, type of reproduction involved. Eplenospore is thin walled, it is formed by asexual reproduction, correct. Zygospore thick walled, it is formed by sexual reproduction, is correct. Germ spore thick walled, it is formed by germ spore, sexual. Germ spore does not show thick wall, it shows very thin wall. So, wall here is wrong. Chlamydospore thin-walled asexual. Asexual is correct, but chlamydospore shows very thick wall. Chlamydospore, how it is formed? Every filament produces septa in the old stages. The total protoplasmic contents fuses alternately, get surrounded by thick wall, result in the formation of chlamydospores. So, position of chlamydospores in Rhizopus mycelium is always intercalary. That means a gap will be there. So, eplanospore thin-walled asexual spore is correct. Zygospore thick-walled sexual spore is correct. Gemspore thick-walled is wrong. Gemspore is always thin-walled. Sexual is correct. Chlamydospore thin-walled asexual is correct, but thin-walled is wrong. Thin-walled is wrong because chlamydospore shows very thick layered wall. So, for this the answer will be eplanospore thin-walled asexual reproduction, zygospore thick-walled sexual reproduction. So, question answer is third one, 1 and 2, 3 and 4 are wrong. In Rhizopus, the diploid structure is zygospore. In Rhizopus, the diploid structure is zygospore is correct because this is the only diploid structure. All the other structures are haploid, that's why the life cycle is called haplontic cycle. Rhizospore has many nuclei in it. Zygospore has many nuclei in it is also correct. Why? Because after the germination, many diploid nuclei undergo reduction division and produce numerous nuclei. So, many haploid nuclei are present, many diploid nuclei are present. But because if it is given as many diploid nuclei in it, first option may be correct. But here it is given, not given whether a diploid nuclei or haploid nuclei. So, second option is correct, but not related to first one. That means Rhizopus, the diploid structure is zygospore is correct. Zygospore has many nuclei in it, correct, but it is not the correct explanation of A. So, here the answer will be A and R are correct. R is not the correct explanation of A. The life cycle of Rhizopus is haplontic, definitely haplontic because diploid zygospore is formed. Zygospore immediately undergoes meiosis to produce haploid nuclei. There is no sporophytic structure in the life history of Rhizopus except zygospore. Zygospore is the only sporophytic. What is sporophytic? Diploid structure. So, zygospore is the only sporophytic structure in Rhizopus. That's why the life cycle of Rhizopus is called haplontic. All the stages coming to homothallism, heterothallism, compatibility, semi-compatible, self-incompatible, formation of eplanospore, formation of chlamydospore, formation of germspore, meiospore, all are haploid. No question of diploid sporophytic stages except zygospore. So, both A and R are correct. R is the correct explanation of A, first option. Rhizopus is heterotrophic. Heterotrophic means it cannot synthesize its own food material because chlorophyll is absent. So, heterotrophic thallophytes are Rhizopus. The reserved food material in Rhizopus is not starch. Whenever they are not autotrophic, no question of chlorophyll, no question of photosynthesis, no chance of reserved food material starch. Because Rhizopus is heterotrophic, it cannot synthesize its own food material due to they are achlorophyllous in nature. The reserved food material in Rhizopus is not starch. So, Rhizopus is heterotrophic, correct. Because it is heterotrophic, the reserved food material is not starch is correct. So, both are related to each other. So, A and R are correct. R is not the correct explanation of A. Gametes of Rhizopus and Spirogyra can be distinguished. Gametes of Rhizopus are cenocytic and those of Spirogyra are uninucleate. Definitely. Gametes of Rhizopus are cenocytic. Why? They are cenogametangia or multigametangia, which ultimately multinucleated structures. Spirogyra gametes are only haploid because the cell contains only one haploid nucleus. So, eplanogametes also contain only one haploid nucleus. So, gametes of Rhizopus and Spirogyra can be distinguished. Definitely, gametes of Rhizopus and Spirogyra can be distinguished. How? The gametes of Rhizopus are cenocytic. Rhizopus itself is cenocytic. Septa formation is completely absent. So, nuclei are irregularly scattered. So, cenocytic is correct. Septa formation may take place in the older hyphae or during reproduction. And those of Spirogyra are uninucleate. Definitely, Spirogyra gametes are uninucleate. So, both are related to each other. Hence, the answer will be both A and R are true. R is the correct explanation of A. Another assertion reasoning. Physical sex differentiation is not found in heterothalism. Physical sex differentiation is not found in sexual heterothalism means a plus mycelium and a minus mycelium come close. They cannot form. So, in the presence of trisporic acid, they converts into zygophores. Zygophores. Here also there is no differentiation. Differentiation comes only when plus and plus come close, they don't fuse. Minus and minus come close, they don't fuse. Plus and minus, plus and plus come close, they don't fuse. Plus and minus come close, only they fuse. So, physical sex differentiation is not found in heterothalism is correct. What is heterothalism? Fusion of two filaments. When plus and minus come close, then only fusion takes place. When plus and plus comes, no fusion. When minus and minus come, no fusion. Physical sex differentiation is not found in heterothalism is 100% correct. All fungi are heterothalic or wrong. Why? Because there are some fungi like Rhizopus sexualis which are homothalic, which complete their life cycle. Plus and minus if they come, they complete their life cycle. Minus and minus they come, they complete their life cycle. So, physical sex differentiation is not found in heterothalism is true. The physical sex differentiation is not found, but physiological sex differentiation is found. What is physiological sex differentiation? Plus mycelium and minus mycelium, then when they come close, then only reproduction takes place. So, that is self-incompatible. So, heterothalism is self-incompatible, homothalism is self-compatible. So, here A is true. Physical sex differentiation is not found in heterothalism is true. But all fungi are heterothalic is wrong. Why? There are some fungi like Rhizopus sexualis which are homothalic in nature. So, third option. Same question repeated again. The Rhizopus sexualis are homothalic species. Definitely Rhizopus sexualis is homothalic species. When two filaments of plus and plus, minus and minus, they can. The gametangia of two hyphae are of the same mycelium, are of the same type. The continuation of this question is Rhizopus sexualis is homothalic species, correct. The gametangia of two hyphae of the same mycelium is correct. So, if the gametangia of the two hyphae of the same mycelium fuse together, it is homothalism. So, Rhizopus sexualis are homothalic species. The gametangia of two hyphae of the same mycelium can fuse together. Question is incomplete, fuse together. Both A and R are correct. R is the correct explanation of A. Thick walled structures of Rhizopus. Thick walled structures of Rhizopus. In Rhizopus, whenever asexual reproduction takes place, eplanospores are formed and chlamydospores are formed. Eplanospores are formed in favorable conditions. Chlamydospores are formed in unfavorable conditions. These are thick walled structures. These are thin walled structures. Thin walled, thick walled. Whenever sexual reproduction takes place, germ spores are formed. Germ spores are meiospores are formed and zygospore is formed. Germ spores and meiospores are thin walled structures, whereas zygospores are very thick walled structures. So, asexual reproduction results in the formation of thin walled structures like eplanospores or sporangiospores, thick walled structures like chlamydospores. Sexual reproduction results in the formation of thin walled structures called germ spores or meiospores, thick walled structures called zygospores, which are surrounded by two layer wall around it. Outer layer is called exospore, exosporium. Inner layer is called endosporium. So, here what are the given? Sporangiospores are given, which are nothing but eplanospores formed during asexual reproduction. Then germ spores are given, which are formed during sexual reproduction. germ spores are formed during sexual. Zygospores are formed during sexual reproduction. They are thick walled structures. So, sporangiospores are thin walled, germ spores are thin walled, zygospores are thick walled. So, for this the answer is 3 only. So, these are some of the important questions we discussed in classification of plant kingdom, Spirogyra and Rhizopus, and these questions are very important regarding all these things, and we have discussed almost all points regarding classification of plant kingdom, Spirogyra, Rhizopus. We have done all the problems related to Spirogyra filaments, calariform conjugation, lateral conjugation and other things, and we have done almost all possible problems even in Rhizopus. What happens if plus mycelium with so many nuclei, minus mycelium with so many nuclei, common fuse, how many diploid nuclei formed, how many haploid nuclei formed, all these things, and almost all concepts we have touched. Next class we discuss the 3 plant groups in plant kingdom, Bryophyta Funaria, Pteridophyta teres and Gymnosperm cycas. All these 3 we discuss in the next class. Okay.